We begin today's program. I request Alice to please take over the proceedings.
Good evening to one and all. I welcome you all to Popular Science webinar lecture organized by Indian Women Scientists Association. I kindly request everyone to rise up for the national anthem. Now I request Ms. Neha Elizabeth Koshi ma'am to come forward and say a few words.
A very warm good afternoon to respective principal, vice principals, IQAC coordinator, our esteemed guest speaker, IWSA members, teaching fraternity, and of course, to all students, the future scientists of tomorrow. On behalf of the Department of Zoology, it gives me great pleasure to welcome all of you to this popular science webinar lecture, Development of Human Embryo, 40 Weeks or 400 Million Years, held in association with IWSA. This is the third popular science lecture that we have conducted in collaboration with them, and their faith and the backing shown by them has indeed renewed the enthusiasm and energy of our entire department. Popular science is defined as an interpretation of science and scientific research developments meant for the general audience. We received an overwhelming number of registrations, more than 360 from all parts of the country. This shows that there are so many like-minded individuals with a yearning to learn more. As Albert Einstein said, the whole of science is nothing more than a refinement of everyday thinking. The field of developmental biology has traveled light years ahead of where it was when Carl Ernst von Baer, way back in 1827, observed the mammalian ovum for the first time on an enlightening journey into the world of embryogenesis. Even though the field of developmental biology has progressed so much, there is still much, much more to observe, see, learn, and understand. I would like to end by quoting Helen Reddy with her lines, which sums up everything. We are all like embryos with a long, long way to go. Thank you.
Thank you so much, ma'am. Now I call forward Ms. Ruchika Parker to introduce our today's esteemed speaker.
Good evening, respected principal, vice principal, honorable guest, and my dear friends. It's a great pleasure for me to introduce our honorable speaker for today, Dr. Srila Janai, an associated professor for IIT Bombay, who is going to talk to us about development of human embryos 40 weeks or 800 million years. It is an interesting topic and I am sure we all are keen to listen and learn about it. Our today guest speaker is an inspiration to our younger generation. She has dedicated 28 years on her life in field of science and research. So quickly walk through her educational background. She has done her bachelor's and master's degrees of science in the field of microbiology from Mumbai University. Further, she has done her junior research fellowship in the field of developmental genetics from Tata Institute of Fundamental Research in Mumbai. And she has also worked as assistant professor. And she also has done research associated in the field of human genetics at Bombay Hospital Institute of Medical Science in Mumbai. She has pursued her Doctor of Philosophy postdoctoral research fellowship in the field of developmental biology from University of California, Irvine, US. Further, she has done her postdoctoral research fellowship in the field of cells and developmental biology from University of Wisconsin, Madison, USA. She served as assistant professor in Department of Biological Science in Tata Institute of Fundamental Research in Mumbai from 2012 to 2020. Currently, she is an associate professor in Department of Bioscience and Bioengineering in Indian Institute of Technology, IIT Bombay. She has done more than 15 research publications in various reputed national and international research journals and also received to various research grant and fellowships like Wellcome Trust Department and Biotechnology, Indian Alliance Intermediate Fellow 2014 to 2020. Science and Engineering Research Board SERB Power Grant 2021 ongoing currently. Science, sorry. She is also been invited as resource person for more than 30 international national conference, although in outreaching program and mentoring of junior research fellowship for masters and PhD students. Apart from being a scientist, she is also known as a Bharatnatyam dancer and sporadical a poet. It's indeed a privilege for me to introduce our guest speaker for today with a warm welcome, Dr. Srila Nair, to take forward it.
Thank you, Ruchika. I'm going to start my screen sharing now. All right. I hope you guys can see the screen. So thank you very much for the very warm welcome. At the outset, I want to thank Susan from the Indian Women Scientists Association who contacted me regarding this public lecture. So I'm really thankful to Susan for giving me this opportunity and for figuring out the logistics with Wilson College and in terms of funding to make this event possible. I also want to thank Wilson College for hosting this talk. Especially Neha, who has been coordinating with Susan and me with respect to all logistics, registration, etc. So thank you to Wilson College as well as Indian Women Scientists Association for having me. It's a pleasure. So I'm going to kind of jump right into the talk. You guys can stop me at any point. I cannot see people. So if you have questions, raise your hands. And I realized that this talk is being live streamed on YouTube as well. So I would be happy to take questions from both fora. So, so today's popular lecture has less to do with development and more to do with evolution. And it is partly because development and evolution are two sides of the same coin. You cannot understand development without understanding a little bit of evolution as well. The more evolution you understand, the better your developmental biology knowledge becomes. So like everybody, when I was an undergrad in India, my undergrad was in microbiology. I had never heard of development or developmental biology. I had done zoology. It was one of my subjects. So it's kind of fitting that, you know, this talk is happening hosted by a zoology department. So developmental biology as a subject was not taught to us during my time as an undergrad in Bombay. I first heard of development when I joined the Tata Institute of Fundamental Research, and I have never looked back from that time. So the pull of this field is quite big. Once people enter it, it's very rare to find them exiting and doing something else. They fall in love with this field for life because it is a beautiful way of looking at science. So I put this title up partly because while I was figuring out what to say today, I realized that a lot of development talks, including mine, focus on certain organ systems. And then we try to understand the development of that organ, let's say either in humans or Drosophila or zebrafish, which are different model systems, or mice. And then we add a few more models here and there to say, okay, the knowledge gap because of this model system is fulfilled by this other model system. So together we have a comprehensive understanding. So today's talk is something I teach in the first part of my developmental biology lecture in a little bit more depth, but I've tried to simplify it for a more general audience today. And the question is, when we look at human development or development of any animal for that matter, should we look at it only in the gestation period for that particular animal? So humans take 40 weeks and zebrafish, for instance, takes 24 hours. Or do we need to also take into context sometimes the entire history till that organism has evolved on planet Earth? So that is a overarching question for today's talk. And I'm going to fill in some bits of interesting information that I think put this question into context for this audience. So as I mentioned, human embryos take approximately 40 weeks to fully develop. And they are typically staged using this staging index called Carnegie stages, CS. And this is what it looks like. And for the most part, by the 8th week, the embryo is referred to as a fetus. And it has all major organ systems, which are already in place: the brain, spinal cord, lungs, heart, everything is in place, kidneys, everything is in place, circulatory system, all of them are in place. They just need to grow and consolidate their connection and mature as organs. So the rest of the gestational time out of these 8 weeks is spent in growing and consolidating these organs. So a lot of action happens during development at very, very early stages. Now, part of this early stage, which is very rarely discussed outside of textbooks and in labs, is the first week right after fertilization. So this is one half of the female reproductive system. And you see here the ovary. I hope you can see my cursor moving. You can see the ovary here. This ovary releases an egg at each menstrual cycle. This egg is taken up by the fallopian tubes, and the sperm actually swim all the way up through the fallopian tubes and reach this egg, fertilizes it, and the fertilized egg sort of tumbles along this fallopian tube till it reaches the uterus, in the lining of which it implants. So this entire process takes about a week. So 7 or 8 days or 6 or 7 days after fertilization by the sperm, the oocyte, the egg completes a certain series of divisions and other events. and ends up in the uterus where it implants and continues to grow along this 40-week period. Now, the important thing to remember is any errors during this very first stage of conception invariably results in loss of that conception, and it's not carried to term. But some errors that occur after implantation can result in birth, maybe with babies that have birth defects. So this period of fertilization is obligately necessary for the system to develop as normal as possible, because otherwise there is going to be no fetus at all. Now, for all of us, I mean, I have a question. This is some questions that I generally have. Like, what does it mean to be human? So you can all write in the chat box, what according to you does it mean to be a human? And we can discuss this at the end today. So for most of us, on a very, very superficial level, it means to be able to do complex tasks. It means to be able to think, because we have the privilege of having the brain, which is a complex, highly organized structure capable of extremely convoluted and complex logic and reasoning. It means to be empathetic. It means to be kind, helpful, and curious. So these are like some of the traits that human beings possess. But aside from all of these, what else does it mean to be a human? So if you ask me as a scientist, this is the answer I would give you. So I would say to be a human on a cellular level means this. So we began as unicellular organisms, and from a unicellular state, we moved into a multicellular state. From a multicellular, so this is basically just division of a single cell. You made a collection of cells. But we are not a loose collection of cells, right? So the cells need to be able to stick together, which means that cells need to develop connections with each other and stay close to each other and in physical contact with each other. So if you shake them or move them, they're not going to just collapse and fall apart. The other thing that these collections of stuck cells need to do is they need to become different from each other. So there is no point in having a collection of cells that are all the same, because then, whether you stay as a single cell or you collect together as multiple cells makes no difference. There has to be division of labor in this collection. So that is provided by some groups of cells taking on functions which are different from their neighbors. So you need a loose collection of diverse cell types which are all stuck together. Okay, but this is also not enough. We are not just a loose collection of cells that are stuck together, which are of diverse types. We are also organized, which means that in that collection of stuck cells of diverse types, there is some degree of spatial organization. So for just representation purposes, I've drawn these, I've shown these yellow cells in the center, these red and purple cells surrounding them on the outside, and the blue, which is the outermost layer surrounding the whole thing as a covering. So you need a multicellular connected state of diverse cell types which are organized. And that, to a scientist like me, is what a human is. And every single system in our body follows this rule. So this module, though it doesn't look like it is a realistic model, this is exactly what our bodies are made of. So there is a skeletal system, there is circulatory system, there is nervous system, the endoderm system, which is the gut and all its associated organ, the digestive system. This is the respiratory systems, for instance. All of these follow this fundamental inviolate rule, which is there is a diversity of cell types. These cells are all stuck with each other, and they are organized in some form where there is a polarity to what is on the inside and what is on the outside. So all animals, not just human beings, all animals, which are called metazoans, are made up of this basic repeating unit. And the cool thing is this fundamental cellular logic, which I schematized just using a bunch of circles, was not invented for animals, and it was certainly not invented only for humans. So this logic has existed much, much before human beings came into existence, or Homo sapiens came into existence. Now, what now the question becomes is, we began as single cells, and somewhere along the line we became multicellular, and then somewhere along the line we became that organized, multicellular, diverse structure. So how did multicellularity actually originate? So multicellularity seems to, there are again several theories for origin of multicellularity. And one of them is that from a unicellular ancestor, we had a multicellular ancestor. And this multicellular ancestor was our common ancestor. So this is known as an LCA, or a last common ancestor. And that last common ancestor could have been multicellular. So, and from there, the three major multicellular lineages diverged. So there were plants, there is fungi, and there is metazoans, which are animals. But this theory is actually not correct, because there is no last common multicellular ancestor for which we. Instead, what evolution seems to have done is, and this is important. So the origin of multicellularity is important, because as soon as animal embryos get fertilized and begin division, they begin life as a multicellular entity. If they cannot become multicellular, they cannot exist. So this is like the base, the fundamental hurdle all animal embryos, regardless of species, have to cross. So evolutionarily, how did animals cross this barrier, or how did evolution cross this barrier, is a very interesting question to answer. Now, what it turns out evolution has done is, there is a unicellular ancestor, and that unicellular ancestor, from that unicellular ancestor, lineages diverged. And during the divergence scenario, multicellularity originated over and over again. So by the last count, multicellularity has independently originated at least 20 times in different lineages across evolution. So I'm only showing 3 of these, which are plant, fungi, and animals. And at the base of each lineage, there is a multicellular ancestor. This multicellular ancestor at the root of the plant lineage is different from the multicellular ancestor at the root of the fungi, which is different from the multicellular ancestor at the root of animals or metazoans. So it means it has evolved multiple times. And this is especially strengthened by experimental evidence, which show that there are no genes to date that have been discovered that we can say, okay, if we express this gene or this collection of genes, we can become multicellular from a unicellular state. So no such genes or groups of genes have been discovered. Instead, common genes control basic processes like replication, synthesis, transcription, which bacteria also possess, prokaryotes also possess. So the fact that common processes for which genes exist are shared with prokaryotes, no collection of genes exists which can dictate or drive multicellularity, and that it has occurred independently many, many times, have led to this current theory that multicellularity has originated independently in animal lineage, in multicellular systems over and over again, because that's a more, let's say, energy efficient, maybe not energy efficient, that's a good way to survive. Now, we will come to why that may be a good way to survive in the next slide or so. So what is, how exactly did this unicellular ancestor become multicellular? And I will also tell you what this unicellular ancestor was. So there are two aspects of multicellularity, or two types of multicellularity. One is called obligate, the other is called facultative. So you need not remember the terminologies. What you need to remember is that obligate multicellularity is essentially clonal. So by clonal means I exist because there was an egg cell that divided, got fertilized and divided over and over and over again. So I am a clone of that original egg cell that got fertilized. So my development and development of everyone in this audience has been clonal. And because of how multicellularity originated, when the original egg cell divided, it did not fall apart. Those cells stayed together and continued to develop more and more multicellular cells. So all animals go through this, and when this process, which is termed as clonal multicellularity, essentially means that there is a bottleneck. So there is a constraint on the system where unless that constraint is met, that multicellular organism cannot come into existence. So for sexually reproducing organisms, it is a formation of a zygote, which is an event of fertilization. So unless the fertilization event happens, the multicellular life form cannot exist. So that is an obligate process. Facultative multicellular systems are essentially loose collections of cells, and then upon certain circumstances, which could be environmental in nature or random changes in their genetic makeup, these will aggregate and they will become multicellular. But as soon as that constraint is gone, that external pressure is gone, they will fall apart and become unicellular again. And some of the, so this aggregative multicellularity or facultative multicellularity has certain advantages in that every time you could get together with different cells, right? But that is inherently disadvantageous also, because sometimes you may like your neighboring cells and sometimes you may not like your neighboring cells. So you may, if you get into conflicts or fights with your neighboring cells, then that aggregate is not a stable aggregate and it will fall apart. So diversity in types of aggregate is an advantage, but the lack of robustness of that aggregate is a disadvantage. But many animals also exist as aggregative multicellulars. But what is interesting to note is that there have been studies where people have looked to see, so clonal means one cell divided over and over and over again to give rise to the entire multicellular animal. Aggregative means different cells got together and made a multicellular cell. So just focus on this panel A. What has been shown by assaying diversity of cell types across many different animals is that the more diverse the cell types, so this is log number of cell types, the more diverse the cell type, the origin of that organism is clonal in nature. So if the general logic is that if you want to evolve complex life forms, so remember that multicolored circle in which I had blue on the outside, purple and red inside and yellow on the very inside. If you want to develop into that form, then you must be undergoing clonal development. So you start with a single cell and that cell divides over and over again. So there is an advantage. Even though there is a bottleneck of fertilization, there is an advantage to clonally developing as a multicellular system. Those systems give you more range of diversity that is possible. So I thought I was actually looking for a logic to put this slide, and then I decided to use my own picture. So this is me, I'm human. And so what does my ancestor look like? So all of you will agree that my ancestor evolutionarily looked like this, right? So I evolutionarily came from these kinds of animals. But we also know scientifically that my ancestor looked like this. Mine and ancestor looked like this. So this green red cell looks nothing like me, but evolutionarily, I owe it to this green red cell to have actually come into existence. Okay. Now this is a logic that I want to convince you of in the next few slides. So what are these green red cells? This is a single cell. It just has different parts. This is a unicellular cell. The chimpanzee and I are multicellular organisms. And I'm saying that, of course, I came from a chimp, but I also did come from this organism, which is a single cell organism. Now, this single cell organism is essentially an animal called choanoflagellate. They are animals. They are not prokaryotes. They are actually animals. And these animals can exist as single cell. This single cell can divide. So remember, to become complex, you have to be able to divide clonally. A single cell has to divide and give rise to daughter cells, and those daughter cells have to stick together. So this single cell can divide. It can divide multiple times, and it can generate what is known as a clone. Okay, and this particular organism can exist in any of these forms. It can exist as single cell or these loose collections of cells. Now, and to be connected with each other, they make physical connections. So there are little bridges that connect cell 1 to cell 2, and then so on and so forth. And on the outside, they deposit certain proteins and other substances which holds them together. So there's like a jelly-like substance which holds them together, which we know from cell biological studies is like an extracellular matrix. So there's a matrix that holds these cells together. And very interestingly, when they are together, they can also form colonies which contract. So these organisms, these animals are responsive to light. And in the movie that is playing on the side here, what you will see is this collection of cells that sort of everts, it turns itself inside out in response to light, and it will collapse back in upon itself. So this property of contractility. So in cells, contractility is driven by a protein machinery called actomyosin network. But this organism, without having a proper actomyosin network, can also contract. This is collective cell behavior that this animal demonstrates. So the very first unicellular animal, was actually unicellular, and its name is it's a choanoflagellate. Now, from choanoflagellates, the next group that evolved are sponges. So sponges are ubiquitous. You will find them when you go diving in any part of the ocean in the world. And they can be in fresh water or seawater. And in this particular experiment, the scientists have placed a dye outside the sponge body. And these guys are filter feeders, right? So whatever is in the water, they will absorb through their holes, their pores. Porifera means those that contain holes. So they'll absorb it through their body, and this pigment comes in, and the sponge, if there was food in it, they would absorb it, and then it comes out through that big mouth opening. So these guys are filter feeders. But interestingly, in these filter feeder animals, so these are now multicellular. Okay, so I hope you remember my original drawing where there was a single cell and then this multicellular structure which stuck together. And then in that multicellular structure which stuck together, you had multicolored cells. So the sponge is one such multicolored, multicellular animal where now you have several different cell types, 4 or 5 or 10 different cell types. So one of the cell, interestingly, is called a choanocyte. And these are pictures of, so the panel A is a picture of a choanoflagellate, that animal, the single-celled animal. And in panel B, you have what are known as choanocytes, which are the cells that are lining the body wall of this animal, which help it absorb nutrition. So the only job of the choanoflagellate animal, the single-celled animal, is to eat. It has a long flagellum. It moves in water using that flagellum, and it collects food inside its body and eats it. The same is true for the choanoflagellate. The only difference being that this is now no longer free floating in water. It is part of a larger collection of cells, and its job in this larger collection of cells is to make sure that that large animal can eat. So the choanoflagellates are, the choanocytes are thought to be the evolutionary adaptation of choanoflagellates because they are very, very similar in structure. They express very similar genes as well. And sponges come in many different varieties. So you had now a single cell, but all the modules that I listed, from a single cell becoming multicellular, getting stuck together, making sure that you had different cells are all going in the same multicellular structure. All of those existed much, much before human beings came into existence. Now, another organism that evolved right after sponges is this organism called Trichoplax. This organism is very abundant in nature, but very poorly. So there are very few studies that describe Trichoplax. And Trichoplax is essentially a two-layered animal. It is like a wafer. It just crawls like an amoeba on a surface, but amoeba is a single-celled animal. But Trichoplax is multicellular. So it has two layers of cells. So you can imagine one set of beads on one surface, one set of beads at the bottom. There is a small slit in between them by which it takes in food. So what this animal does, so this moves using these cilia. So in this movie here, you can see the cilia. So it sort of glides on the cilia on the surface. So from its body, cilia are projecting. These projecting cilia touch the bottom of its surface, and then it sort of glides along the surface. While it is gliding, it releases from its gland cell, it releases certain enzymes which digest food in the environment, and the digested lysed food is absorbed by this animal. And when it is absorbing it in that space between these two layers. So now, from just being a collection of cells, so sponges have like a single layered existence, you have now made two layers. You have a layer on the top. So the top, this layer, this animal has a bottom which is touching the surface, and a top which is above, right, where what we are seeing. So it now has a bottom and a top, and it has now made a two-layered structure. And this animal is known to have 6 different cell types. So the types of those cells are not important. What is important is that you have now begun in evolution the module that you want to use for more evolved animals. So you went from single cell to multi-cells. Those multi-cells stuck together, they adopted different fates. Now those different fated cells are adopting an organization. So you are deciding that, okay, I'm going to have gland cells in these positions. I'm going to have cilia projecting at the bottom, but not so much at the top. So you're entering into an organizational mode using these diverse cell types. So once you have set up these 3 or 4 different modules in earlier events of evolution, the next thing you want is an ability to sense and react to your environment, which is done by the nervous system. And again, the nervous system did not originate in higher evolved animals or later evolved animals. It originated very quickly in comb jellies. So comb jellies and another group of variants to which Hydra belongs are possess something known as a nerve net. So a nerve net is essentially this mesh of cells which are present right beneath the surface. And when you poke this animal, it will go away. If it touches something that it doesn't like, it will react and move away. So it has a rudimentary nervous system, and it feeds by taking in entirety animals, small animals from its environment, which is usually water. Now, they are called comb jellies because you can see these birefringent structures, these things, these flaps that are moving. These are like little combs, and they keep continuously moving, both to propel the organism and to ward off others from its immediate environment. So comb jellies are one of the first instances where a nerve net-like structure has been documented. So much before the brain evolved, much before a proper nervous system evolved, a system existed which would allow an animal to sense and respond to its natural environment. And that could be a form of a predator. So this comb jelly, the one it is tumbling, so what it does is it has these long flagella, 2 long flagella that you saw. It traps food using these long flagella, and then it retracts these flagella inside its body cavity. And when it retracts, along with the flagella, it also eats whatever was stuck to the flagella. These are very, very beautiful to look. Now, once you've made a nervous system, one of the other creatures to which we owe our ancestry are cnidarians. And cnidarians are special.
Body is a hollow column with wall.
Sorry. Cnidarians are special because they are like many animals of this particular genus. They are able to regenerate indefinitely. So there's a joke that if you want to study regeneration or aging, for instance, not regeneration, aging, Hydra is a bad animal to choose because Hydra never ages. Every cell in its body is replaced at 20-day interval. So it has this continuously renewing stem cell population, which keeps adding new cells over and over again. So it's able to regenerate very robustly. And what you will see is it reproduces by budding. It can also reproduce by sexual reproduction. And there have been experiments that people have done where this entire animal has been blended into a soup. And this soup, when you leave that soup alone, which is essentially a single cell, so that was a Hydra that budded off from the mother Hydra. When you blend this soup of and leave it naturally, cells in this soup will aggregate with each other and it will re-establish its body plan. So I'll discuss the Hydra body plan in a little bit detail in the next slide. But essentially, these animals have two layers. There is an outer layer and then there is an inner layer. And what is going to happen is if you take a Hydra and cut it into half, each of these halves will regrow into a full animal. So it can completely, the tail part will grow a head and the head part will grow a tail. So the molecular signals, which is Wnt based, that is required to make a head or make a tail is very well established. So this is the, and like Hydra, there are many other animals that are capable of regenerating body parts extremely faithfully. Okay, and somewhere along the line, we have lost that ability to regenerate robustly. So it's not like we completely lost it, but certain parts of our bodies can. So this is the, the outer layer in pink and the inner layer in green. It has a mouth, and this is the soup in which it was blended. So such animals, labeled animals, were just mashed up into single cells. And if you observe this, it's remarkable that these cells sort of self-sort themselves, and they will immediately know which ones go on the outside, which ones stay on the inside. And not only do they do this inside out organization, they also do the head tail organization. They know from where to make the head, from where to make the tail. And in a few days, it's as if you have never cut this animal. It just completely regrows back into its original shape, and it will continue to reproduce and make more Hydra, and it's perfectly well. So it's like virtually indestructible because it regenerates very robustly. Now, this animal, the Hydra, like the comb jellies, also has a nerve net, a rudimentary nerve net, which is this collection of cells that are spread like a spider web, which allows this animal to react and respond to its environment. So what I have said up until now is you need to be multicellular. You need to have diverse cell types. They need to be organized, and preferably you need a nervous system. But none of these examples that I showed you is a human being or an animal or a complex animal. So what is missing? So despite having all of these required modules, what is missing? So if you compare the events up until now with that week 1 event of human embryonic development or human post-fertilization life, you will find many similarities with this. So cell division, DNA replication, sticking to each other, diversifying cell type, making inside versus outside. All of those will be found in these systems and in common with that week 1 of a fertilized embryo's life. But it is still not a human. So a ball of cells is not a human. So what is missing? So what is missing is again, an organizational logistic, which is humans are a group of animals, metazoans, which have a 3-layered body plan, which means that they have an ectoderm and an endoderm. So that green and pink cells in the Hydra, one of that is ectoderm, the outer one is ectoderm, and the green one, which was inside, is endoderm. And in between, you need a 3rd layer, which is a mesoderm that Hydra does not possess. So Hydra does not have a mesoderm. And all the examples that I showed up until Hydra, the quanoflagellate, the sponges, the ctenophores, cnidaria, none of those guys have a mesoderm. They all have a 2-layered body. So you need a 3rd layer. And that 3rd layer is the mesoderm. And the mesoderm also gave rise to a very unique structure in development called the notochord. And that notochord, the presence of that notochord, is why we are classified as chordates. So insects are not chordates, we are chordates, right? Insects do have a mesoderm though, but we additionally have used that mesoderm to make a structure called the notochord. And the reason this having this notochord or having mesoderm is important because, so again, I go back to my simple schematics. I hope it makes some sense to you, but I did not want to use complicated, dev bio examples. So we start with this basic 2-layered body plan where some of these cells can be differently colored so that you have a diversity of cell types. And you can imagine this blue and yellow ball to be that ball of cells in week 1 of human embryonic development. Okay. Now from this ball of cells, you need to convert it into a 3-layered structure. So you have an inside layer and an outside layer, but you need a layer in between. And that in-between layer is created by this process called gastrulation. And that in-between cell type that is created is the mesoderm. Okay. And once you make a mesoderm, we are not spherical animals. We are elongated animals. We have a front or a top-down axis, right? So we are long in one dimension. So that length in one dimension is comes into existence because of a movement called convergent extension, where cells will converge towards each other. And when they are converging towards each other, they will elongate in the opposite direction. So convergence in one axis, elongation in a 90-degree perpendicular axis is where this elongation process happens. So literally during convergent extension, because of the notochord and mesoderm, the body axis gets pulled and stretched lengthwise. So you have a notochord. All animals have a notochord during embryonic stages. There are very, very few animals that retain a notochord in the adult life. So right now, none of us have a notochord. The notochord disappeared sometime during embryonic development. It gives rise to a very special cell type in the back of the body. But the presence of that notochord, what it allowed the surrounding cells to do is, wherever there is mesoderm, it signals to the overlying ectoderm to pattern itself into certain structures. So you can imagine that at the top of the head, the mesoderm allowed the brain, the cranial muscles, all of those to come into existence. And as you went down the body, down the head to tail axis of the body, it came into contact with the mouth parts, the endoderm, which will give rise to the stomach, liver, pancreas, the coiling of the gut. And the mesoderm itself made organs. So the heart, for instance, is made of mesoderm. The kidneys is made of mesoderm. The musculature is made of mesoderm. The entire skeletal system is made of mesoderm. So the evolution of this, or the presence of this one cell type, allowed the other cell types that were in contact with it to adopt many, many, many different kinds of fate. So if you look at organ development and look at the evolutionary traces of how many organs come from ectoderm, which is one of the cell layers, and how many organs come from the endoderm, which is the other cell layer. These two are the original primitive layers. You know, even Hydra has it, ctenophores have it, all of these animals have it. Versus if you look at all these fancy organs that we have, most of those fancy organs come because of the mesoderm. They are made up of mesodermal cells, or they came from other organs because that other organ, the other cell type, was in contact with the mesoderm. So in development, in evolution and development of animals, the ability to make mesoderm is absolutely critical to being a complex animal. But the interesting thing is, so during gastrulation, the physical cell emerges, right? So all of this, in theory, this discussion is okay. But you need a cell, you need a physical cell to come. So that is what this process of gastrulation does. And I will not go into gastrulation today. That deserves a lecture in and of itself, maybe some other time. But gastrulation is where sheets, these loose balls of cells will, twist and turn and move with each other to make one outer layer, one middle layer, and one inner layer. And that 3-layered body plan is required to further enhance the complexity. Otherwise, complexity cannot emerge. But the interesting thing is, so though the mesoderm can do all of this, the origin of the cell type called mesoderm can do all of this for the animal in which that event occurred. The genes that mesoderm cells express are present even in animals without a mesoderm or a notochord. So for instance, there are a group of genes called T-box genes. They are transcription factors, especially genes like brachyury. So I won't again go into names of these genes, but what I want you to understand is that the quanoflagellate, which is that single-celled ancestor, does not contain any of these T-box transcription factors. Okay. On the other hand, the sponge, which is the porifera, the ctenophore, which is the comb jelly, or the cnidarian, which is the hydra, all possess different numbers of genes to make mesoderm tissue. Though, remember, they do not have mesoderm. But the gene to make mesoderm-like cells exist in them. And of course, bilaterians have the biggest diversity of these T-box genes, which is up to 30 genes of various different T boxes exist. T box essentially is a DNA binding domain. Because of that protein domain, they are classified as T box transcription factors. So it is interesting that even in the two-layered organisms like Porifera, ctenophores, cnidarians, which do not possess a mesoderm-like genes or transcription factor-like genes are actually expressed. But this theme is not unique to mesoderm. In fact, if you look at choanoflagellates, so these are papers, more recent papers from Nicole King's lab and some from others, where they have shown that all of the genes, so proteins are divided into different types of families based on functions that they do. So what is seen is that proteins that are required for addition, for extracellular matrix formation, for, I'm trying to remove my, proteins that are required for extracellular matrix attachment, or for regulation by transcription factor, or for cell-to-cell signaling. So when cells are stuck with each other, they need to talk to each other. Otherwise, there is no point in being together. So that is usually achieved by receptor ligand signaling. There is something expressed by one cell, which the other cell can read and understand and respond to that. So that is signaling, that's called cell signaling. So these cell-to-cell signaling event, proteins for all of these processes and more are present in choanoflagellates also. And so of course, they do not have the entire intact protein, but elements like domains of these proteins are present in the choanoflagellate protein collection as well. So for instance, these are different kinds of ligands. So some of these you may be familiar with. This is a transcription factor based cell signaling pathway. This is a ligand notch delta based signaling pathway. This is a small molecule based signaling pathway. This is a kinase based signaling pathway, RTK. All of these are pathways which we use rampantly. So every single organ system in the body requires one or more, if not all of these pathways at some point in time during development and homeostasis. The choanoflagellate, which looks nothing like us, still has all of these and more during its evolutionary timescale. So the model that, the viewpoint that we are arriving at is the basic bricks of becoming multicellular existed in a unicellular ancestor. It also existed in a multicellular ancestor, which was not very complicated. It was a very simple multicellular ancestor. And then over time, especially as new cell layers began to develop, like the mesoderm developed, the mesoderm began to move and stretch. Then those modules which were already present evolutionarily got used to diversify and consolidate the complexity, which which began much before in time. So the distance between a choanoflagellate, the evolutionary distance between a choanoflagellate and a human is approximately 800 million years, which is the reason why I gave the title the way I did. And I also would like to, so I'm going to end my talk now because I would like to ask, take questions. I also want you all to be able to go and look at a couple of websites. So I'm going to stop this particular screen sharing. So in the timeline of human evolution, we evolved somewhere here in the Ediacaran period. We evolved in the sense multicellular life forms evolved somewhere around this period. And then of course, this is where we are currently. So there's a very neat app. It's also it's on a website, and it's also available as an app, since you're a generation that's likely to use information if it is present in an app. So I'm going to show you, not as an app, but on the website, what this time tree website looks like. And I encourage everyone to go and take a look at it. So let me share my internet screen. So this is what the time tree interface looks like. On the app, it is much more simplified, but you can always come back here because the full-fledged website has a lot more information. So you can type human, for instance. You can see my screen, no? And if I type Porifera, which is a search that I did earlier, it will tell you what is the evolutionary distance, how many millions of years apart is a sponge from a human, okay? And all of these are indicators or data. So this is a extremely, it's a very well cross-referenced database, and I spend quite a bit of time sometimes on this. So it tells you what the carbon dioxide levels were, what the oxygen levels were, what is the era or period that you are in, and how many millions of years away from the presence of humans did that event occur. So if you go back and instead of, let me see, if I typed, I think I tried to look for quinoflagellate, but I was not able to find them. So let's see how different we are from chimps. So from chimps, we are 6.4 million years apart. And if you zoom in, so the reason I wanted to zoom in is because this also tells you which were the verified Earth impact events. So impact data is essentially when a meteorite struck Earth, where it struck. So these are verified impacts. And there have been dozens of, like not dozens, it's like a huge number of impacts that the Earth has gone through, some of which has also resulted in extinction of entire species. So this is a useful website and an interesting website, even if you're not keen on the science behind it. Just as a curiosity exercise, this is a useful website to go through. They also have several resources where you can look at animal diversity, you can look at the catalog of life, the catalog of diversity of fish, the diversity of genome, diversity on the basis of nucleotide, which is DNA collection, or on the basis of protein, and PubMed, which is the database where all this raw information is deposited. The other resource I want to share is this website called onezoom.org. And here also you can look at, again, let's say I don't want to look at humans, but I want to look at Drosophila, which is another model system that is popular. So any one of these. So this essentially lists where the species is, and you can zoom out. So if you scroll down the screen, you can zoom out and it will tell you from, so this is where the species I searched for was there. And it tells you where in the tree of life, how far apart from a common ancestor does this species, so this is the branch where we were. So this tree is again, extremely complicated, extremely rich and very, very diverse. And it tells you, so I was somewhere in this branch over here. And what is the evolutionary distance? What are the different ways in which, what are the different paths this evolution needed to take in order to get to where it was. So it will come all the way down to eukaryotes and prokaryotes. So sometimes it can be a little disconcerting, but you can look at this over and over again. So I wanted to go back to my original state, but it's going to take us some time to scroll back in time. But this also tells you that where we are. So, it's not a joke to travel 800 million years in a tree like this. So you guys can use this information. So we were somewhere along this route. So this is the original tree, right? This composes of everything that we know of, from eukaryotes to plants to metazoans, everywhere. And you can scroll through these trees. So it tells you all of life, where archaea, where all other animals are placed within this, amphibians, mammals, all of them are part of this tree. And this is also fairly updated on a regular basis. So both of these resources are very useful resources if you're interested in understanding development in an evolutionary context. So I'm going to stop my talk and end by thanking everybody for listening. Thank you again for having me, and I'm happy to take questions.
Thank you so much, ma'am, for such an inspirational and educative talk. Thank you so much for guiding us with such and enlightening us with such informational wisdom of yours. I'm so sorry. The questions would be taken after a presentation that is going to be.
Oh, sure. Sure.
So much, ma'am. Now I request Dr. Shamala Bharadwaj ma'am, the Vice President of IWSA, to come forward and illuminate us with more into this topic.
Good evening, everyone. Am I audible?
Yes.
Yes, ma'am.
Okay, I'm sorry. I am coming in between the very interesting talk and the question answer session. But this is because I was not given the chance in the beginning before.
Oh, sorry.
But anyway, bear with me. I would just like to share some of the information about IWSA in this forum for the benefit of the students who have listened to this talk. Let me just share my screen. Are you able to see my screen?
Yes.
Okay, so let me briefly tell about Indian Women Scientists Association, which is a non-political and a non-profit organization, which was started by 12 eminent scientists. You can see way back in 1973, and we are going to complete 50 years in another 2 years. And so that is the slogan for the 50-year celebration, looking back to look forward and all. And we have been, and at present, here are the 5 board of trustee members who guide us to take forward whatever was the vision of these 12 founder members. So with this, I just briefly tell you, we are engaged in several community welfare activities and educational activities as well. We have a working women hostel, a nursery school to take care of young children, daycare center, computer center, health care center, library for the students. Then we also run a diploma course for the early childhood care education connected with this nursery school and daycare center, which is in the 25th year running, and advanced learning center for computers for various students. And we also have several scholarships and awards for the students. The students should take note of it and visit our website. They will come to know what are the scholarships and awards which we have installed for the college and college students and others. Okay, and our main focus is on how to restore the ecosystem. We have several green initiatives for which I don't have time to discuss now, but I am just giving you the glimpse, and you can get more information from our website, how we are they have taken up several green initiatives like solar systems, biogas plants, and several others. So more information, please look into our website. So this is our hostel building. See, girls and women who come from all parts of India, they have a safe haven to stay here, a 160 room hostel where they can safely stay and go for their work or study. And during the pandemic, we have followed the protocol very strictly. And this is how this hostel is going on. Of course, with less capacity now due to pandemic. And we have a beautiful learning garden. I invite you after the pandemic is all over to come and visit our headquarters at Navi Mumbai, Vashi, and you will see so much of flora and fauna around our in our center, where this will give a very good learning experience. Like today, Dr. Srilaja explained about all the evolution of the cells from this. The plants also have their own evolution, I think so. And then we have a healthcare center where we have a dental chair, and also some physiotherapist helps the all nearby citizens to take care of their health, and we provide health facilities and carry out some cancer detection camps and so on. This is our health center activity. And we, as I told you, among our green initiatives is the solar energy system to generate electricity as well as for heating water for the hospitals. And we have a new collaborative program with Vigyan Prasad. It is called Each One Teach One, where a college student acts as a mentor for a school student on one-to-one basis. So the college student also kind of improves her understanding and communication skills, and the young school student also is very kind of, she is more at home with like a elder sibling and learns the lessons for interest and all. So this is one initiative which is going on right now. And we have the other initiatives which have started, internships for the college students. We have started a science club for the school students, and then several other online programs are going on. The pandemic has not dampened our spirits. We are going ahead with several online programs during this pandemic, which we were carrying out physically earlier. So we have several new lecture series besides the one now today you heard, the BRNS supported popular science lecture for the students. We have other lecture series also going on. And as I told you, successfully we completed 2 internship programs with students of SIS Nerul and Jai Hind College, Mumbai. More about it you can see in our website, and write to us if you are interested in joining our intern program. This is where several projects were taken up with Jai Hind College during June and July this year. And our mouthpiece is it's a newsletter which we were bringing out 3 issues per year till last year. Now we are bringing 4 issues per year, and this you can easily download from our website. You can read about our activities, and also I forgot to mention we have 11 branches all over India. So they also are engaged in similar activities. You can know about the activities of headquarters as well as branches from the newsletter, and also read articles of interest, the current scientific interest in these newsletters. And the computer center, as I told you, is have several programs of various computer skill developments for students as well as seniors. And the computer center is celebrating 30 years of its completion. You can, if you want, you can join this webinar, which is on 26th September. Please see our website, and then you will know if you are interested in joining this celebration of 30 years of our computer center. Very interesting seminar is being organized by our the chairman of our board of trustees, Dr. Sunita Mahajan. So with this, I just stop my presentation. I hope you would have got a glance of about our activities. Now, please carry on with your question answers, which will be more interesting for students to know more from Dr. Srilaja. Thank you very much.
Thank you so much, ma'am, for guiding us and showing us how inspiring the association is. Now we would like to take the questions.
So how do we do this? Do I just...
Ma'am, there are a few questions in the chat box. So do you want me to read it to you?
Yes, please. That would be nice. Thank you.
So the question is from Avita Kohli. If more than one hydra are blended, will the cells still regrow to form a single hydra? Which part of the mixture would preferably develop head?
So if you mix two hydra, which is exactly what was done. So in that blending experiment, more than one hydra was originally blended, and you can, as many drops as you put, you will get a particular sized hydra. If you put a large drop, you could get a large hydra. And in that mixture of cells, where the head comes depends upon where wind signaling tends to become more active. So if you have high levels of wind signaling, that structure will become a head. If you have low levels of wind signaling, that structure tends to become the tail. But yes, the size of the organism can depend upon how many cells are part of that original soup.
The next question is from Jyoti Ramchandani. Are the T box genes silent in organisms that lack mesoderm?
So that's a good question, because the current data is based on the presence of the gene in the genome of the organism. We don't know whether they are expressed in terms of protein or RNA. So in choanoflagellates, it's known that the genes for ECM and the other proteins domains like Wnt or hedgehog, those are expressed, but because they are not full hedgehog proteins, they contain elements of the hedgehog protein or the Wnt protein or this RTK signaling receptor. It may not function like the way we know these ligands and receptors to function. So both happens. Either they are present and maybe not expressed, but in general in evolution, if something is not expressed, it's eliminated. If that gene exists, it means that it is used sometime during the life of that organism. So my guess would be there is some data for expression studies. These are called RNA expression studies. So some data exists for all of these species for many of these genes. But if they were not being expressed, then evolution would have eliminated them. So they are expressed.
The next question is from Ananya Sachdev. The immediate question, of course, is why conoflagellates have proteins similar to cell-signaling proteins. How does one explain their presence?
So choanoflagellates also need to stick to each other. So the minute two cells will stick to each other, they need to know, am I surrounded by 2 cells or 4 cells, or am I alone? So for that, communication is necessary. And sometimes communication is also necessary to inform other or cells within that collection what is the status of the environment. So therefore, you need cells to be able to talk to each other. Right, all the cells may decide to go in one direction. So in that movie where you saw that sheet of cells, folding outside, folding inside out, somewhere there is a trigger point, right? Somewhere the cells in that group. So if you have a straight sheet of cells, for that sheet to bend, the cells at that bend have to change shape. So that is a collective decision. So if cells cannot talk to each other, then this sheet will never change shape. So communication is necessary for any kind of coordinated activity. It is like running a 3-legged or a multi-legged race. If you don't coordinate with your neighbors, you're not going to be able to do anything. So if cells don't talk, the movement that you saw in that video for choanoflagellate, you will not see it, because cells don't know that they need to be together. They need to work together to achieve that movement. So communication, if more than one entity is together, they must communicate for that interaction to be meaningful. Did that answer the question?
Yes.
Well, at least that's my viewpoint.
I would also like to let people know, if anyone wants to unmute themselves and ask questions, they can do that too. The next question is by Nisha. I want to ask about evolution of human beings, sapiens sapiens and Cro-Magnon man brain comparison.
Sorry, Alice, I missed the question. I couldn't understand.
Ma'am, it says the comparison between Homo sapiens sapiens and brain capacity?
Oh, I won't. I mean, I cannot comment on that because the answer is I don't know. I'm sure the human brain, the Homo sapien brain capacity is better because the Cro-Magnon man doesn't exist. We do.
The next question is by Yogesh Dixit. What is the genetic basis of fluorescence shown by some organisms?
So, so some of them are fluorescent proteins. So GFP was discovered like that. It is naturally present in a jellyfish. So organisms express proteins that are capable of being green or red color in nature. And oftentimes for butterflies, for instance, it's not like they express, so they are called pigments. So there is a whole variety of pigments that animals can express. But the ctenophores, for instance, when they are the ctenophores, for instance, that comb jelly which you saw, reflecting in multicolors, sometimes they take in food which is multicolored from their environment, or sometimes they have crystals that are placed or structures that are placed that when light falls and reflects, it gives a prism-like effect and gives a colorful halo to the whole animal. So there is genetically encoded proteins that are fluorescent like GFP, and there are non-fluorescent pigments as well, which have a biosynthetic pathway. Just like plant makes pigmented flowers, so do animals. And then there are ways in which light can be reflected off of a surface, which birds and butterflies and animals use to appear colored. Depends upon the angle you look at them in the third case.
Ms. Shagun Sharma, do you have a question? You have raised your hand.
Yes, I have a question.
Yes, go ahead.
Good afternoon, good evening, ma'am.
Hi.
My question is, can the chimerism is an example of allocative multicellularity?
Sorry, could you repeat it? Your voice broke in between.
Okay. Ma'am, chimerism, the ghost twins, have you heard about it? And can be an example of allocative multicellularity?
No. So even chimerism, the way you are describing it, it comes from a clone. So when cells are propagating in a clone, some cells become different from the others, and therefore you get chimera. But it is not aggregation of cells. Cells, like our cells, don't aggregate just like that. So you can chimeras. So we make chimeras, we make mosaics, for instance, in the lab. So where you can mix a GFP labeled zebrafish with a M cherry or a red labeled zebrafish, and you can generate a chimera, but they are still zebrafish cells that mix together with each other. To a limited extent, non-zebrafish cells can also get incorporated, but those chimeras need to be put inside that animal very, very early in development. But eukaryotic cells in general tend to stick. So they are sticky because of extracellular matrix components, they will stick to each other. But the aggregative mechanism of forming a multicellular structure is different from the way the chimera integrate with each other.
Okay. Thank you, ma'am.
Ma'am, the next question is by Ananya Sachdev. Why are genes expressed in notochord present also in animals that don't have notochords?
Yeah, so see, why is a very difficult question to answer. We only know it exists. In many of these organisms, so typically, to understand what a gene does, scientists have typically knocked out those genes. You delete those genes, or you make that function, you abrogate that function, and then you can test, in the absence of that gene, how does the animal develop. So that is the basis of all genetic studies, and much of the information we know about organ development or development in general, animal development in general. But many of these species, like the choanoflagellate or the ctenophore, the comb jellies are not jellyfish, by the way. They're a completely different genus of animals. In those animals, they are not model organisms. They are organisms for whom we simply have sequence data. So you need to develop technologies to knock out genes in a quanoflagellate or a ctenophore, grow them, and then see in the absence of those genes, what does the animal look like, or does the animal survive? Only then can one answer, why are these genes expressed? What do they do in the quanoflagellate or ctenophore? We don't know that, because knockouts in these models don't exist.
So even without the knockout experiments, we don't yet have any hypothesis, popular hypothesis for why.
Our hypothesis is based on our own work, right? Like what they do in higher animals or more evolved animals. But these earlier evolved animals do not have those structures. So then the question is, in the absence of these structures, which we know on the basis of experiments, these genes control, what else are they doing? That answer we can only know once we knock them out.
Okay, thank you.
The next question is from Naman. Is there any role of cilia in developmental signaling?
Yes, of course. So hedgehog, for instance, one of the proteins that I just discussed in one of the slides, it is known to be transported to the tips of the cilia, and cilia act as signaling centers. Cilia do many things. They are required to move things from one place to the other. They can concentrate things. So the ciliary movement is what decides left-right axis in many organisms. Cilia can also be used as signaling beacon. So because they project outside of the cell, they can, you can put, the cell can put things at the tip and signal to other cells that, oh, look here, I have this protein expressed on me. So cilia is absolutely required for all kinds of signaling processes.
Thank you so much, ma'am.
I think, Alice, there is one last question.
I'll take questions if they are from the YouTube channel.
Yes, we have pasted those as well here.
Okay, cool.
There's a question from Alok Yadav. Are we close enough to make a whole organ or organism through stem cells by understanding different evolutionary studies?
So are we close enough? So we can make cell types in a petri dish. So you can make a liver cell type or a lung or a brain or a kidney or a pancreas in a dish, but it doesn't look like the organ. So the 3D shape of that organ is not there. It's still a loose collection of cells. So there are different ways in which scientists are trying to not just make the cells take on a particular fate, but for those collection of cells to take on a particular shape. So the pancreas has a particular shape, the lung, liver, heart, all have a particular shape. And for organs to function properly, they need to also have a proper 3D shape. So one way is to provide them with a template of an extracellular matrix and then seed these cells, which are grown as stem cells, into that matrix, the extracellular matrix, and hope that that organ becomes fully functional. So that is being done. But beyond that, we are not, I mean, those are some of the efforts that I can think of. There are also efforts where organoids are being grown. So if you can force an organoid, so you take cells from an adult, put it in culture, and you don't need to now go through the stem cell route. You can just have cells, divide multiple times, provided certain factors are given to them. And in that organoid model, you can try to coax cells to take on, or organs to take on a particular shape by maybe giving them a template. So cells need a template. Collections of cells need a template to take on a 3D shape. But the interesting question is that during development, in an embryo, no organ had a template. Just collections of cells self-organized into a 3D shape for that organ. So when we are studying it in a culture system, like a stem cell culture system, how do you spontaneously make organs take 3D shape is a challenge, and there are no easy answers for that yet. Providing a template of an ECM is one example, but otherwise there are no easy answers yet.
So have scientists thought of this idea that instead of growing the organs and organoids in culture, studying how the embryos develop in the mother's womb can be imitate the same thing inside a human body?
But how do you do that? So we do that. This is exactly what developmental biologists do. So my lab studies zebrafish development. So we know how the zebrafish heart develops. It's a two-chambered heart. When you go beyond zebrafish into mouse, the animal becomes little opaque. You cannot see through the body. Humans, though, it's a far-fetched thing. You're never going to be able to see it unless some rules and regulations actually change. So there's a ban on using human embryos beyond day 14, which recently got relaxed. The other alternative is scientists have made in 2021, there are groups that have made human monkey chimeric embryos. And in using those chimeric embryos, you may be, because they're not fully human, you may be able to culture such chimeric embryos in an in vitro lab-like setting. And in that embryo, you can watch how organs are developing and then use those principles in a stem cell or cell culture system. That's a possibility. But the human monkey chimera work is quite controversial. All of these are going to be controversial, but they are happening. They happened as recent as 2020 to 2021, but groups are doing that.
Thank you, ma'am.
Yeah.
Today's last question is by Vikrant Burde. What controls programming during development gene expression or signaling?
That's a very broad question. So everything is controlled. So at the very beginning, there are many tiers of control. So one is which genes get expressed. Which genes get expressed largely depends upon epigenetic marks that are present on that DNA, histone marks that are present, how the DNA is folded. Then even if the DNA is unfolded, are there the required transcription factors to sit on that segment and transcribe the gene? So gene expression is a multi-tiered complex process, starting from epigenetics all the way through to transcription and translation. Even after the RNA gets transcribed or the protein gets translated, the proteins themselves can get modified. And once it's modified, it can either be functional or become non-functional or be targeted for degradation. All of that put together will result in an output of function. So gene expression is fairly complicated. It's controlled by multiple tiers of regulation.
Thank you so much, ma'am, for enlightening us with such important knowledge and wisdom. That is all with the questions.
All right.
Now I request Mr. Anas Khan to propose the vote of thanks.
So, a very good evening to one and all present here. It is my privilege to deliver a vote of thanks for this event. So, on behalf of Wilson College and the entire audience present here, I extend a very heartfelt thanks to our guest speaker, Dr. Srilaja Nair. Thank you for enlightening us about the development of human embryos. How does the multicellular organisms originated from unicellular ones? And the line that really amazed me was that the genes to form multicellular organisms were already in the unicellular organisms. So thank you for enlightening us with such knowledge. I would like to express my deep gratitude to IWSA, that is Indian Women Scientists Association, President Dr. Rita Mukhopadhyay, Vice President Dr. Shyamala Bharadwaj, Program Advisor Dr. Suzanne Ipen, and USA representatives, and BRNS, that is Board of Research in Nuclear Sciences, for collaborating with the Department of Zoology, Wilson College, in organizing this webinar. We would also like to acknowledge and thank the principal, the vice principal, IQAC coordinator, and Department of Zoology of Wilson College. I would like to thank all the volunteers for their efforts in helping and to organize their webinar. And last but not the least, we thank all the participants from all across the country for actively participating in today's session. We thank you for being with us this evening. This brings us to the end of the program. So thank you. The
Thank you,
link for the
Thank you so
feedback
much.
form is already
Thank you very much.
already pasted in the Google Meet, in the Zoom app, as well as on the WhatsApp group. So you can fill it in half an hour, and the certificates will be sent by email on the email address that you provide.
Thank you, everyone. Bye.
Thank you, Srilaja. Thank you, Sneha, and all the others.
Thank you so much. It was a wonderful session. Thank you.
Thanks. Bye. Bye, Susan.
Bye bye.
Thank you, ma'am.